So Maximilian Strobl is another of our colleagues here at Moffitt. He's also with the University of Oxford, and he is working with Sandy Anderson and Philip Maney on many things, including what he's going to be presenting today. He's about to get, I think, a doctorate in mathematical biology, and I know that there is at least one experimentalist at Moffitt who is waiting for him to come and finish some experiments. So I'm hoping to see him back in Moffitt in a few months. So Maxi, I know that you've done a lot of work on evolutionary enlightened treatments, and I'm looking forward to see what you present today.
Thanks, David, for the very kind introduction. Yeah, so my plan is to really continue this discussion, which has been really interesting on the role of spatial in adaptive therapy. And specifically, I think what I want to talk about is that I think it's not only going to be sensitive cells we need to think about, but also the competition between resistant cells. And that's something I think that we're missing when you're looking at non-spatial models, and that's something that comes out when you start looking at spatial models. But before I go there, I just want to say this is something that we made as from an OD model that will be part of this talk. It's not super relevant, but I thought it was kind of nice and festive, so I included it on the cover slide. What I want to think about and talk about today is sort of the selection criteria for adaptive therapy. When should we treat adaptively, and when should we treat with more intensive or aggressive approaches? And it's something we've kind of reviewed yesterday, and Yannick had a really nice summary. And I would say sort of some of the key factors that have emerged so far are obviously the resistance fraction. The less there is present, the longer we can contain it. And then also this idea of vicinity to carrying capacity, that the more constrained the growth of this population is, the better we'll be able to leverage that constraint in competition. And then also, I mean, this idea of a fitness cost, which is something that Yannick has kind of pointed out, and that's come up as well this morning a little bit. And then one point as well, which is something that we kind of looked at earlier in my PhD, is the role of turnover. And specifically, one of the things we did is we kind of built a very simple ODE model where we had two populations and sort of assumed just Lotka-Volterra competition dynamics. And one of the things we found was actually that the role of turnover is quite important, because it essentially determines the speed of evolution within the tissue. And so it decides how quickly you're going to be shifting back from resistant to sensitive, and for example, what impact a resistant cell cost will have. And so this is now out in cancer research. And one of the things that kind of came up, and many of you who know Sandy won't be surprised, is the next question was so what does space do? How about that? How does it change our conclusions? And so what we thought is we've had spatial models in adaptive therapy, and that being said, they've always been kind of qualitatively quite different to the non-spatial models. And so we thought, let's actually build a spatial model that's very close to the non-spatial model we've just analyzed, and compare the two and see where are they similar and where do they differ. So what can we learn from a spatial model that we can't necessarily see in the non-spatial model? And so what I did is essentially I took this very simple non-spatial model and turned it into an agent-based model. And so we're still assuming that we have a simple binary type of resistance where we have sensitive cells and we've got resistant cells, and that we have a drug that kills only dividing sensitive cells, and that these cells live on a lattice. And then what we would do is we will continue compare continuous treatment at some kind of MTD to the adaptive protocol from the prostate trial. And we parameterize this using parameters from prostate cancer. And here's kind of just sort of an overview of what the model looks like, and get you acquainted with the graphs and sort of terminology I will be using. So here on the left-hand side, what you can see is first I've drawn the graph for continuous treatment, and you can see how we kind of get this very classical competitive release, where initially we have a very good response, but then after some time it grows back, and it's mainly made up of resistant cells. And if you look at the images from the simulations, that's exactly what is happening as well. In contrast, if we do adaptive therapy on the system, you can see that we can actually go for a number of cycles before we see eventual progression. And that happens later than the continuous setting. So in this case, we do get a benefit from adaptive therapy. And that's interesting, actually, because one of the things as well I decided to do was to not start with the kind of more circular spheroid-like models we've seen so far, but actually seed these cells randomly, because I felt that would be a little bit more of a worst case analysis. Because obviously if the resistant cells are trapped in the center, it'll be easy to contain them. But what if it's more heterogeneous? Can we still control it then? And one of the things that this simulation here suggests is that even in that case, we still have a chance. So one of the things I then did was I took this model and analyzed some of the trends we had seen in the non-spatial model. And so, for example, as I said, one of the factors was that the proximity to carrying capacity and the resistance fraction are important. And that's something that's also true for this model. So on the x-axis here is the proximity to carrying capacity of being, so kind of how densely we seed the cells. And then we've got two different initial resistance fractions. And you can see that the smaller resistance fraction and the kind of denser the tissue initially, the better it is. Also, to cost and turnover have kind of a similar interaction again. So if I don't have any turnover in the tissue, then if I had a resistance cost here on the proliferation, not that much happens. In contrast, if I have turnover in the tissue, then here in the purple line, you can see how gradually we're getting more and more of a gain from adaptive therapy. Cool. So far about the qualitative agreement. I mean, all of this is very nice and comforting because it means the ODE models are useful for getting a good understanding of what's going on. What about quantitative comparisons? So what I did is I went back and took the ODE model, and I used the same sort of microscale parameters in the ODE and the agent-based model. So that means that the replication rate of an individual cell would be the same as sort of in the exponential sense in the ODE and the spatial model. And what I did is I then compared the simulations that I would get. And what I found was that, not surprisingly perhaps, there were quite significant quantitative differences. So here on the top is the simulation that we were just looking at with now the two overlaid. So I'm just showing the adaptive trajectory here and the progression of that here in yellow. And then under here is the sort of corresponding ODE simulation. And you can see that there's this, in this case, while both predict that adaptive therapy is beneficial, we see very different times of progression, both for the continuous and for the adaptive therapy. And that's something that kind of continues as we map out the space. So for example, here what I'm showing you is the time to progression for adaptive therapy in the ODE model, and that same thing in the ABM. And generally speaking, what we find is that when it comes to absolute time to progression in the system, that tends to be larger in the agent-based model than in the non-spatial model. That being said, if we look kind of at how much do we gain relative to continuous therapy, because as you can see, obviously also the continuous therapy has changed, then here it seems like the ODE is perhaps slightly overestimating, or at least kind of being more optimistic about the gain than the agent-based model, because generally speaking, we see less relative gain in the agent-based model than in the ODE, which might also be due to the fact that I'm seeding these randomly. So what is going on? How can we explain some of these discrepancies? And so one of the things to answer that I did was I looked at the simulations in more detail. For example, if we do that here, one of the things that we can see is that kind of as we become more similar between the two models, the defining features is the number of resistant cells at the beginning. And really with that, what changes is not only that number, but also sort of how they look like in space. And we go from these really sort of small patches that cause the growth up here to these large, sort of much more diffuse, almost carpet-like structures down at the bottom. And that makes sense that causes a difference in the quantitative predictions, because obviously up here we're going to have a much more sort of much smaller surface to volume ratio for the resistant population than we will have down here, where a lot of the resistant cells are exposed to open space to divide into. And so I think one of the things going forward that we need to think about is how do we accurately actually model the growth of this resistant population, given it's going to be dependent on the spatial spread. Something else I also kind of points out though is that one of the differences that we see is how these cells compete and interact with each other. And that made us wonder, what does competition actually look like in that adaptive therapy? Because so far we've always talked about it, and we've kind of maybe seen some experimental evidence for it, but it's difficult to show this in experiments. So we thought we've got an ABM here, we've got a perfect system, we can actually measure this. So what does this actually look like here? And so what I did is there's two types of measurements. The first thing I looked at was how much competition is there, which I quantified by looking at how many resistant cell divisions are blocked over time. And then when I do that, I get a result that looks as follows. So here, yellow and blue again, yellow is continuous and blue is adaptive therapy. And then on the y-axis here is the percentage of blocking that we see. And what we can see is that whenever we take drug off, as we would expect, that blocking goes up. So here we can really see adaptive therapy in action. That being said, what is also kind of curious about this is that generally speaking, the rate of inhibition of the resistant population goes up. It does so as well in the continuous therapy, till at the end it actually seems to overlap that of the adaptive therapy. And that's kind of curious. And initially it wasn't clear to me what was going on. But then actually, if you look at the images and you wonder, and you ask, who are they actually competing with? You get a picture like as follows. So what I've done here is I've looked at what proportion of the neighborhood of a cell, a resistant cell, is made up of either resistant or sensitive cells. And what we can see is that actually most of these cells are resistant. So quite a large proportion in the simulation of the resistant cells are actually competing with other resistant cells. And I think actually that's quite, it's subtle, but I think it's quite important. Because if the paradigm is competition with sensitive cells is what is the benefit here, then for example, if we take a resistant population and we kind of tear it apart, then the further we tear it apart, the more exposure we essentially have to a sensitive population. So you'd think that is what should maximize the benefit of adaptive therapy. But actually, when I let these grow out and then look at the benefit of adaptive therapy, that's not the case. It is the most benefit is here, where we actually have both intraspecific and interspecific competition. And so I think one of the points for me is kind of thinking about competition in more detail, and I've adapted this diagram from Carl O'Malley, is that yes, we have competition between sensitive and resistant cells, but I think we also have competition between resistant cells. And I think that is relevant to think about when it comes to thinking about how should we modulate therapy. Because I think, for example, if we have gradients of resistant, then we might want to modulate therapy to use sort of less resistant cells at the edge of a colony to contain the more resistant ones at the core. Finally, I just want to briefly kind of show some interesting results we got from looking at some data and asking, does any of this have anything to do with reality? And so what we did is there's data available, which is probably familiar to some of you, from an intermittent trial on prostate cancer. And the data is available for over 60 patients. And again, it's androgen-deprivation therapy, and it's given intermittently. And it's not too dissimilar actually to the adaptive therapy trial, except that here treatment is given according to an absolute threshold. So it's not a relative decrease, but an absolute decrease. And it's given in kind of fixed lengths of time that the patients receive. And one of the things that kind of jumped out at us when analyzing this data was that there's different speeds of cycling in these patients. And so we see, for example, here a patient that cycles very quickly, and here one that cycles much more slowly. So we asked, can our ABM tell us anything about these patients? And so what I did is I took the ABM and I let the 4 parameters that have kind of been varying so far free and try to fit it to these data. And what I found is that, interesting enough, actually I can fit this data, which I was surprised and quite impressed by. But not only that, when I then asked what are the parameters really that are driving the response here, what we found was that the key parameters really seem to be cost and turnover. If I fit just the initial conditions, it doesn't work. But if I fit cost and turnover, I can obtain fits which are almost as good as the ones I showed you previously. And so these appear now with just 2 patient-specific parameters. What is interesting as well is if I plot these 2 parameters against each other in sort of patient space, so each of these points here is a patient, we see a trade-off between the 2. So we see them kind of cluster along a specific line in parameter space. And moreover, as we kind of walk along that line, it seems like the patients the cycling is accelerating. So it's kind of caused by this interaction between cost and turnover. And the final point that I was kind of curious about this is if we then look at the simulations sort of pictorially that are associated with that, we see that in these fast cycling patients, we have a much more sort of carpet-like structure, as I was kind of talking about earlier, than in these very slow cycling patients. And what is kind of curious is if you go back to the patient, back to the trial, one of the things they found was that patients with a Gleason score of less than 6 had a much longer sort of, or tended to have longer off-cycling times than patients with a higher Gleason score. And so that makes us think that maybe here we have some evidence for higher Gleason, and here we have some evidence for maybe lower Gleason patients. I think it also jives quite well with what Jill was just showing in her previous talk. So where next, I think one point for me is we should really think more carefully about spatial structure and how to incorporate that into the models. Again, I've been using kind of very rough structures here. I also think we should think about rhesus competition in more detail. Here I've used space, but obviously there's other types of competition. And I think that might change how exactly adaptive therapy acts. It works or does not work. And I think also the role of normal tissue should obviously be looked at. And I think here also what is kind of interesting is connecting models of tumor evolution back to the models we're actually using for adaptive therapy. And so that brings me to the end here. I hope I've shown you that I think we should not only be thinking about competition with sensitive cells, but also competition with resistant cells, which means that we need to think about where and how often resistance arises. And I think that's where, for example, the mode in which it arises, whether it's genetic or environmental, I think will be quite important. Then also sort of kind of connecting back to Jill's talk, I think we should be thinking about what can we learn from cycling dynamics about how we should be adapting treatment. And just to conclude, I want to thank all my collaborators and mentors on this project and throughout my PhD. And here, just sort of the QR codes to some of the papers that have come out of this. Thank you very much.
Cool. So we have not as much time this time for questions because we have to move on to the next talk in just 2 minutes. But I'll start just with Heiko, who says, Maxi, great talk. As for the ABM fit to the Rzyszowski data, are the three parameters cost and turnover uniquely identifiable? What conclusions can you draw for the biology of evolution?
So I've not done a sort of formal sensitivity analysis, but from my experience, the convergence is typically quite good. I don't get multiple optima or have problems with my fitting algorithm. So I think you have a decent hold of them. In terms of what they actually tell us about the biology, I'm not totally sure yet. I think what they might be reflecting, for example, if the turnover is different underlying biology in terms of the structure of the tissue and potentially also, for example, the impact of the immune system. But I think actually I'm just going to say I don't know, and I think that's something I would like to kind of think more about going forward. Cool.
Arne Tralson asks, there are quite a few degrees of freedom you have when you implement the spatial agent-based model, but typically only one of the OD. How robust is your comparison of the OD with the agent-based model?
From my understanding, I would say in terms of the qualitative comparison, so for example, what parameters do and how they interact, I think it's reasonably robust. I think the one thing that would change things is a mutation rate. Mm-hmm. Quantitatively, I think he's absolutely right. It depends very much on the details of the implementation. And so, for example, I think if we took this off lattice, then we would probably again see, I think it would be qualitatively similar, but obviously sort of how much they quantitatively differ, I think would change again.
Michael Ratz asks if there is a linear relationship between cost and turnover, does fitting only one of them while fixing the other also work?
I found so just fixing the other on a population level does not work. I've not tried sort of, for example, exploiting the using a linear relationship to then fit the other. I've not tried that, but I would guess that should work given it's such a strong correlation.
So we have time for one more question. I know that Hites asked as well a question, but then he later points out that that's the question that Heiko asked before, so we're going to skip that. And then Fred asks, Fred Adler, Ergan presentation and approach, I might have missed this, but Do your models include asymmetric competitive effects, as we heard about yesterday? This speaks to the issue of where costs are realized, where in growth, death, or competition.
No is the answer for this particular work. This is just looking at the growth rate. In the non-spatial paper, we do look at other types of costs, and I agree with him very much, it matters, but in this context, I've only looked at growth rate costs. One final thing I maybe just wanted to say for Heiko's question, I think it's sort of, I think the closest relationship to biology that I found so far is sort of this relationship back to Gleason score that I can, and tissue structure, as I presented. I think that's where it could correlate.
Cool. So we have more questions, and maybe we can use one of the breaks to have Maxi answer some of them. But in order to keep things going, and knowing that Carlo Males is going to be the presenter now, I think it's probably best if we move on. Thank you very much for your presentation, Maxi.
Thank you.